Hello dear students, welcome to my class. This is Dr. Varsha and you are watching Botany Varsha, the reign of botany. In my last lecture, we have already discussed introductory part of photosynthesis. In today's lecture, we will study mechanism of photosynthesis. Let's start. Photosynthesis. The photosynthesis is differentiated into two parts, oxygenic and anoxygenic photosynthesis. As the name indicates, oxygenic photosynthesis in which oxygen is produced. But in anoxygenic photosynthesis, oxygen is not produced. Examples of oxygenic photosynthesis, green plants, cyanobacteria and algae. And when we talk about the photosynthesis, that means generally we are talking about oxygenic photosynthesis in which oxygen is released. In anoxygenic photosynthesis, examples are purple bacteria and green sulfur bacteria. These bacteria uses electron donors other than water. In oxygenic photosynthesis, green plants generally perform this function and oxygen is released from water after the process of photooxidation of water. But purple bacteria and green sulfur bacteria, they utilize some other electron donors. Photosynthesis is divided into two steps, light dependent reaction or light reaction. In light dependent reaction, light is necessary. This step cannot be completed without lights. That is why it is called light dependent reaction. In this process, assimilatory power is produced in the form of ATP and NADPH. ATP is adenosine triphosphate and NADPH is reduced form of nicotinamide, adenine, dinucleotide, phosphate. And next step is light independent reaction. It is also called dark reaction because light is not required in this process. And in this process, fixation of carbon dioxide is found, and after fixation of CO2, organic food is produced in the form of carbohydrate. They do not require light, but the product of light reaction like assimilatory power ATP and NADPH is utilized in the production of carbohydrate. There are so many evidences in the support of light reaction. First evidence is flashing light experiment. It was given by Warburg in the year 1919. He worked on chlorella plant and he took two sets of green alga chlorella. On one set of chlorella he gave continuous light and on another set of chlorella he gave intermittent light in which light period was followed by dark period. But total period of light was same in both the experiment. He observed yield was different in both the cases. Yield was enhanced in second experiment in which he gave intermittent light. He concluded that the rate of photosynthesis was found to be greater in intermittent light as compared to the continuous light. It indicates the existence of two parts of photosynthesis, light reaction and dark reaction. This was the first experiment in which he gave continuous light, and it was second experiment on chlorella plant in which he gave light period followed by dark period. So alternated periods of light and dark reaction was given to the chlorella plant and he observed the yield was enhanced in the case of intermittent light. So he concluded that there is two steps in light reaction, dark and two steps in photosynthesis, light reaction and dark reaction. Second experiment was given by Robert Hill in the year 1939. Robert Hill was actually worked on Stellaria media plant. He isolated the chloroplast from Stellaria media plant, and that isolated chloroplast was illuminated in the sunlight or some other light similar to the sunlight in cell extract in the presence of water but in the absence of carbon dioxide. And he found that water release oxygen in the absence of CO2. And when he replaced the cell extract by some other iron like ferric oxalates, and during this reaction ferric oxalate is reduced to ferrous oxalate while H2O is oxidized into oxygen. The substance which acts as H2 acceptor is called Hill reagent. Hill reaction confirm that oxygen is produced from photooxidation of water, and it is separate from CO2 fixation for oxygen release. Light is required, so Hill reaction can be also described as light dependent reaction. Now you can see this is chloroplast, and this chloroplast is isolated chloroplast from the Stellaria media plant, and it is illuminated in the presence of sunlight or some other light. In the presence of water, water release oxygen, and water release oxygen from in the absence of carbon dioxide. He also observed that in the absence of carbon dioxide, water released oxygen. And this hydrogen acceptor, oxidized electron acceptor, get reduced during this process. And he also observed when this cell extract is replaced by some other ferric salts, this ferric salts is converted into ferrous salts and act as hydrogen acceptor. So it get reduced, and this is called Hill reagent. So after Hill reaction, it is concluded that oxygen is released from water, not from the sun, not from the carbon dioxide, and because carbon dioxide is absent over here. And in second conclusion was, and in this reaction light is necessary for the production of oxygen, so it is also called light dependent reaction. And this is redox reaction. Oxidation reduction reactions are found over here. So these two reaction actually proves that photosynthesis process is completed in two steps: light reaction and dark reaction. So the existence of two pigment system came from studies made by Robert Emerson and his co-workers called Emerson's effect. Next experiment is Emerson effect. Emerson in the year 1957, and when he was working on chlorella plant, chlorella is an alga, and nowadays it is used as protein and mineral supplements mostly by astronauts. And so Emerson with his co-workers, he was measuring quantum yield of photosynthesis in chlorella. Quantum yield can be calculated by number of oxygen molecules evolved during photosynthesis, and it is divided by amount of different wavelength of light absorbed during this process. So it is called quantum yield. When they were working on this experiment and they were measuring quantum yield of chlorella plant, and he plotted a graph between the light absorbed and quantum yield, and he gave monochromatic light to the chlorella plant. He found the yield was constant up to 660 to 680 680 nanometer, but after some time he found some sharp depression, sharp sudden fall in the red region. Now you can see this graph. This is quantum yield and different wavelength, and the graph was constant, but at 480 nanometer he found a little dip due to the presence of carotenoid pigments, and the graph was enhanced, and after some time when he reached to, and when the quantum yield reached to the 640 to 680 nanometer, beyond 680 nanometer he found a sudden drop in the red region. So sudden drop in the red region in photosynthetic yield, and that in the red region, that drop is called red drop or Emerson first experiment. And in second experiment, Emerson's second experiment, he extended his previous experiment in the year 1958, and he supplied additional shorter wavelength of light along with the longer wavelength of light, because monochromatic light was insufficient in the previous experiment. When he supplemented with shorter wavelength of light, he found enhancement in the rate of photosynthesis and recovered the red drop. This is called enhancement in Emerson's enhancement effect. And actually when he made the graph and rate of the photosynthesis, and rate of the photosynthesis, when he gave 680 nanometer red light to the monochromatic light, and he found that the yield was 52 quantum yield. And when he gave to the chlorella plant 700 nanometer monochromatic light, and the quantum yield was 10. And when he gave both the nanometer, both the lights, red light and far red light, that means 680 nanometer light and 700 nanometer light together, he found drastic enhancement in the yield of photosynthesis, or in quantum yield. This is called Emerson enhancement effect. Next is pigment system. Pigment system, we can also say pigment protein complex, or pigment system 1, or photosystem 1 and photosystem 2. So each photosystem consists of core complex and light harvesting complex, that is also called antenna complex. And the core complex is consisted of reaction center, electron donor, electron acceptor, and light harvesting complex consists of different type of photosynthetic pigment like chlorophyll A, chlorophyll B, and carotenes, xanthophyll, etc. So each pigment system consists of core complex and antenna complex. And core complex, in the core complex, electron donor, electron acceptor, and in antenna complex different type of pigments are found. These pigments are keratin, xanthophyll, and different type of photosynthetic pigments. They take light from the sunlight. They take energy from the sunlight, and this energy is transferred to the reaction center, and this reaction center perform the photochemical act. So pigment system is of two types, pigment system 1 and pigment system 2. In pigment system 1, in both the pigment system, core complex and antenna complex both are found, but reaction center is changed. And in pigment system 1, reaction center is P700, whereas in reaction center in pigment system 2, reaction center is P680. Now you can see the location of pigment system. 1 and pigment system 2. This is the structure of chloroplast. You can see the thylakoid membrane. Chloroplast has two type of thylakoids, stroma and granum thylakoids. Granum thylakoids in which lamelles are arranged one above the other and form a stalk-like structure. These are granum thylakoid, and in granum thylakoid, now in granum thylakoid, this is the structure of granum thylakoid. You can see the location of PS1 and PS2 in the thylakoid membrane. This is the thylakoid membrane, and in the center portion, this space is called lumen, and the outer side is called stroma. This is stroma, and the wall of the thylakoid membrane is consist of biomembrane, that is consisted of lipid bilayer. So in lipid bilayer, pigment system 1, pigment system 2 and ATP synthase are attached. Now the difference between pigment system 1 and pigment system 2. Pigment system 1, in pigment system 1, photooxidation of water is not found, and in pigment system 2, photooxidation of water is found. Actually, after photooxidation of water, oxygen is released. So in pigment system 1, oxygen is not released, but in pigment system 2, oxygen is evolved. Reaction center of PS1 is 700. That means they can absorb light more than 700 nanometer. And P stands for pigments, and reaction center, pigment system 2 reaction center is P680. That means chlorophyll A molecule can absorb less than 680 nanometer light, and the molecular oxygen is not evolved in this system, whereas it is evolved in pigment system 2. And when we talk about the location of the pigment system 1 and 2, pigment system 1 is located on the outer surface of the thylakoid membrane, and pigment system 2 is located on the inner surface of the thylakoid membrane. And pigment system 1 produce NADP, or you can say that NADP is reduced to NADPH in pigment system 1. But in pigment system 2, NADPH is not produced, but electron is donated from the pigment system 2 to pigment system 1 for the reduction of NADP. And pigment system 1 is involved in both cyclic and non-cyclic photophosphorylation, whereas pigment system 2 is involved only in non-cyclic photophosphorylation. And reaction center, both the pigment system consist of pigment and protein. They are also called pigment protein complex. So protein is always found over here. So reaction center is consisted of protein. In PS1, FeS, ferrous sulfur protein is found, but in pigment system 2, reaction center containing quinone and manganese and chlorine. They help in the oxidation of water, and water oxidizing enzyme is also found. And the major function of pigment system 1 is synthesis of NADPH, which works as assimilatory power, and in the production of carbohydrate, the energy is required, which is fulfilled by NADPH and ATP. And pigment system 2 produce ATP and help in the photooxidation of water and release oxygen. Okay, thank you so much. That's all for today's lecture. Thank you for being with me, and stay tuned.